diabetes is a huge problem right now. And using stem cells, scientists and researchers have found a way that we could potentially cure diabetes. And I find that super interesting and cool. So what happens in diabetes is that the cells actually develop a condition called insulin resistance. The pancreas produces insulin, but these cells don't cooperate. And what that results in is glucose accumulating in their blood, which then puts the person at risk for heart disease, nerve damage, etc. But that's where insulin injections come in. However, insulin injections aren't that good for your body, and they have a lot of side effects that are definitely not good for people, especially with diabetes. And so one way that stem cells have a way of curing, almost treating diabetes, is to transplant beta cells. And what beta cells do is that they sense the blood sugar levels and then produce insulin to reduce them. So what would this do for the two different types of diabetes, and how would that help? Type 1 diabetes, the new beta cells would replace the ones they've lost with disease. And for type 2 diabetes, it could increase the body's ability to produce insulin, which then lowers the blood sugar. And so these embryonic stem cells can be then turned into insulin-producing beta cells. But however, there is one challenge that researchers are trying to look into, because the problem is that they're not able to replicate the exact signals that transform as stem cells into beta cells. And it's kind of hard to do, and right now they're not really sure how to do it, which is a potential obstacle in this field. A second way scientists are looking to treat diabetes using stem cells is through stem cells and islet cells. And this treatment is more for people with type 1 diabetes, because people with type 1 diabetes, their beta cells are destroyed so that insulin is not produced at all. And so what are islet cells? Islet cells are essentially just clusters of cells that can be found scattered throughout the pancreas. And then these cells produce and release hormones that then help regulate blood sugar. The majority of these are beta cells, which produce insulin to get glucose into other cells. Islet cell transplants have been around for more than 40 years actually, in animals too. However, most of the time, these new cells don't last very long, since they are very delicate and require a lot of oxygen and blood. However, scientists have figured out a way to supply this oxygen that these islet cells need temporarily by basically engineering a material that releases a lot of oxygen. Another problem that scientists are facing, however, is the question of will the immune system destroy and reject these new cells, which is called transplant rejection. A possible idea to prevent this, however, is to find a way to coat the islet cells with protection from immune attack without blocking their insulin production, instead of just relying on immune suppressing drugs. The third way that stem cells have found to be possible in treating diabetes is using stem cells to and engineering them into the 3 main human cell types that are key components of type 1 diabetes. The 3 types are beta cells, the immune cells that attack and destroy beta cells, and then thymus cells, which educate the immune system by providing information about which cells to destroy or not. And these cells will be produced from patient-derived iPS cells. And using these 3 cells, scientists and researchers are finding a way to almost make a type 1 diabetes animal model that we can then look at for reference to a human model. And so how this will work is that researchers will transplant the type 1 diabetes cell type into the mouse, which will then create a accurate research model for researchers. The new model will help these researchers and scientists test whether cells from a patient generate type 1 diabetes in every case, or whether there are certain factors or environmental conditions that help develop type 1 diabetes. It also shows that if all patients with type 1 diabetes have the same disease, or there are different variations of the disease or like subtypes of the disease. However, in order to do this, researchers must look at the different ways they can grow cells. So the first one is direct differentiation, which can be done by turning pancreatic digestive cells in the live mouse into insulin-producing beta cells. And this can also be done by using a virus to make genetic modifications that could not be safely done in humans. Another method is reprogramming, where cells are taken from patients with diabetes, and then using reprogramming methods, induced pluripotent stem cells are differentiated into beta cells. The third type would be stem cells the self-regulation, where beta cells can make more of themselves. So as of right now, beta cells in the pancreas do produce more of themselves, but very slowly, and this replication slows down even further with age. So there is a lot of hope for people with diabetes because stem cells offers almost 3 ways to treat diabetes as of now, and scientists are looking at different other ways that they could potentially treat in the future. And this will definitely be something that's super interesting, and this is definitely something that's super interesting to me, and will have a huge impact in the future.